page. Welcome to Deschutes Public Library's virtual programming. I'm part of the Deschutes County Library, and this is our wonderful presentation, Endemic Animals of Madagascar. Our presenter tonight is Andrea Baden, PhD. She's Associate Professor of Anthropology at Hunter College of the City University of New York. She is a biological anthropologist with training in primate behavioral and molecular ecology. Her research takes an interdisciplinary approach to answer broad evolutionary questions about lemur adaptation, evolution, and conservation. Andrea, thank you so much for being here tonight. Welcome.
And thanks to all of you for coming to hear me speak with you this evening. And what I'd like to do is take this opportunity today to introduce you to some of the work that's going on in my lab investigating molecular ecology in lemurs, one of Madagascar's endemic animal species, and how we can turn what seems to be a conservation crisis into an opportunity. So broadly speaking, I consider myself a primate behaviorist with research interests in the evolution and maintenance of complex social behaviors, with a focus on fission-fusion social dynamics and cooperative infant care. However, when investigating larger evolutionary questions such as these, I think it's really important to take both an integrative and a collaborative approach. And as such, my research combines traditional methods in primate behavioral ecology, molecular ecology, and conservation biology to investigate aspects of primate social systems that are otherwise not easily detected by observation alone. So for instance, using molecular genetic techniques, we can infer things like patterns of dispersal and gene flow among groups or species populations. This information then allows us to estimate underlying patterns of genetic relatedness or kinship among members within a social group or among social communities. And then paired with methods in behavioral ecology, this allows us to investigate the underpinnings and/or outcomes of diverse social behaviors, including aggression, affiliation, and cooperation. Now, research in my lab focuses primarily, though not exclusively, on the lemuriform primates, and these are commonly referred to as Madagascar's lemurs. This is a taxonomically diverse group of animals. So there are 5 families, more than 103 different species. You can see this down here at the bottom in this cladogram. And they're relatively ancient clades. So these animals evolved anywhere from about 55 to 75 million years ago in Madagascar. They're found no place else in the world, and they've been evolving in relative isolation ever since. And so we tend to think of these animals as being sort of this primitive behavioral state of primates. And this allows us this really sort of unique and unusual and exciting opportunity to investigate the earliest emergence of what it means to be a primate, and then also allows us a comparative opportunity to look at what it means to be a human. So to this end, I've established and maintained a research site in southeastern Madagascar. So this is home to the Ranamafana ruffed lemur project, where I've been working since 2005. And I also run and maintain the Primate Molecular Ecology Lab at Hunter College on the Upper East Side of Manhattan. And together, in collaboration with ecologists, endocrinologists, nutritionists, bioinformaticians, all of these different kinds of scientists, members of my lab are able to use multiple lines of evidence to better understand the behavior, ecology, and evolution of Madagascar's lemurs. And so this is just to illustrate a few of the different fields in which the work in my lab is sort of intersectional. Now, despite all of this, so, we're studying population genetics, landscape genetics, infant care, development of the gut microbiome. Despite all of this diversity, the research that's going on in my lab really does, it's aimed at 2 overarching research questions. And these are, first, how do landscape genetic or landscape features influence patterns of distribution, migration, and genetic diversity in wild primate populations? And how the resulting patterns of genetic relatedness or kinship can help us to explain the evolution and maintenance of complex social behaviors. Now, this is a career's worth of work. And so today I'm only going to focus on the first of these 2 research questions, looking at gene flow, genetic diversity, and how we can use this information to help solve some of these conservation problems. So as I've said, my work concentrates on the lemuriform primates, or the lemurs, and this radiation, along with 3 other mammalian radiations represented here. So you've got the lemurs on the top, the tenrecs here, the nesamine rodents, and the euplurids. These are all endemic to Madagascar, which means they're found in Madagascar and they're found no place else in the world. And Madagascar is located, it's about 400 kilometers, or roughly 250 miles, off the southeastern coast of Africa. And so to put this into perspective, it's about the size of Texas, or roughly 3.5 times the size of Florida, for instance. And so it's this really small landmass, but with this really, really rich biodiversity. And so this island continent has been separated from the rest of mainland Africa for 85 million years. And so all of the plants and animals that are evolving there have been evolving in relative isolation ever since it split off from mainland Africa. Now, unfortunately, despite all of this really amazing biodiversity, it's also considered one of the world's 36 biodiversity hotspots. And that seems, sounds like that might be a really great thing. Unfortunately, though, what this means is that this area has really high biodiversity, but that biodiversity is being threatened with imminent extinction. And this is largely due to deforestation and habitat loss. And so what you can see here in this figure on your left is forest cover in 1953, and then to present day, so present day being 2014. And you can see this both, let me just scooch this up a tiny bit, on the west coast and in the east coast of Madagascar. And so if you see down here on this map, this is the forest cover in Madagascar back in the 50s. Anything that's in green is forest, and as you get warmer in color, that actually indicates forest loss. And so you can see that, by zooming in, doesn't really matter where in Madagascar you are, most of the forest that was there, not like 70 years ago, is pretty much gone at this point. And this is largely due to the fact that rainforest soils in Madagascar are really poor quality. And so you can only really farm those soils for a couple of years before you have to let them lay fallow. You can return to them after a bit of time, and then after that, you pretty much have to just leave them. Nothing will grow from that point forward. And so this leads to further and further encroachment in order for local communities to be able to feed themselves, to feed their families. And so this is really, threatening the endemic flora and fauna, and even the local human populations as this encroachment continues. In fact, there's a recent paper that we just published looking at the fate of Madagascar's rain habitat. And we estimate, based on current deforestation rates, that is assuming that deforestation does not escalate in the future, it remains constant, all of the eastern rainforest corridor in Madagascar should be gone by about 2080. And so this is just to show you under 2 different rainforest cover scenarios, if we assume here on the right that protected areas, so national parks, if we assume those protected areas actually protect forest, and that only forests outside of the protected areas will disappear, then literally by 2080, the only forest that will be left in Madagascar are these protected areas. Unfortunately, we also know that in Madagascar and in lots of other parts of the world, including our own, protection maybe isn't always effective. And so if we relax forest protection, that is, we assume that protected areas don't always protect the forests in the way that we would hope, then by 2080, pretty much everything has disappeared. And so in light of these ongoing challenges, one primary aim in my lab is to investigate how these and other anthropogenic pressures impact patterns of migration and gene flow in lemurs. So why is that even important, right? Why do we care about gene flow and migration in these animals, or any animals? And it really all kind of comes back to our intro to bio classes that we took in high school or college, right? So if we think back to our intro to bio classes, we know gene flow is an important mechanism of evolution that helps to not only maintain genetic diversity within populations, but also by maintaining genetic similarity between populations, right? So essentially, an animal reaches reproductive maturity, disperses out of its group to avoid mating with its relatives, and enters a new group and reproduces, thereby maintaining that genetic diversity and gene flow among these different populations. We also know that when gene flow is interrupted, so in this case by natural occurrences, then gene flow slows, and these different populations will gradually through time accumulate genetic changes that in classic examples can eventually result in speciation events. However, these are generally pretty gradual, pretty slow, allows animals to adapt to their new habitats. But what I'm more interested in are these anthropogenic barriers to gene flow. Okay, so these happen much more rapidly, and this often leads to very rapid declines in population size, something known as a bottleneck event, where essentially you start with this large, genetically diverse population. The population size crashes. For instance, just by chance, if you have a small population here, a large one here, anyone left over here is going to have a much reduced size, a much reduced genetic diversity. And this can lead to populations being susceptible to reduced genetic diversity, which then can affect their long-term potential to adapt and cope with things like climate change or disease. Okay. In addition, depending on an organism's body size, its locomotor preferences, you can see here this poor sloth trying to cross this open area habitat, and also dietary specializations. These different factors all play in together to alter dispersal patterns by creating essentially island habitats across these different altered landscapes. And so for the first part of my talk then, I'm going to describe how black and white ruffed lemurs respond to habitat fragmentation in light of all of this disturbance. And I would like to note really briefly that while I'm focusing on ruffed lemurs today, members of my lab are currently working on similar studies in a whole bunch of different lemurs and other primates worldwide. Okay, so let's talk a little bit about ruffed lemurs. So ruffed lemurs are one of Madagascar's endemic animals. They comprise two species within the genus Varecia. They're moderately sized, so they're 3 to 4 kilos, so like 7 to 8 pounds. They're about the size of a large house cat. They're arboreal, which means they live in the trees. They're obligate frugivores, which means they rely really heavily on fruit, and their digestive tracts and their dentition are all adapted to this high level of frugivory. And they're also really, really highly social. So they live in this is big for lemurs, so large groups. They have fission-fusion dynamics such that animals are sort of like atoms bouncing around in space. They'll come together, form large groups, and then throughout the course of the day, group size and composition changes. And they're also really territorial. So they defend these large territories against other communities or other intruders. Now, what's especially important in terms of conservation is that these animals are widespread. So the red ruffed lemurs here are found in northern Madagascar, and the black and white ruffed lemurs are found throughout the eastern rainforest corridor. So they exist the entire length of the island. However, they're very patchily distributed, and their distribution is really hard to predict. So we don't entirely know why they occur in some areas, but not others. And we think this has to do partly with being highly frugivorous. They're very sensitive to habitat degradation, but they are also the tastiest lemurs, unfortunately. So they eat so much fruit, it makes them really sweet, and so they get targeted. So it's possible they've been hunted out of some of these areas. But all of this together makes them really useful as an indicator species, right? Because they're super sensitive to habitat disturbance. And so if you enter a forest and there's no ruffed lemurs, then it's indicative of forest health being relatively low. But all of this together has led them to be these critically endangered animals that at present we're estimating will be extinct within the next 30 years unless patterns change. So, in order to study their genetic diversity and gene flow, this takes several different phases of research. So the first of these is to characterize the genetic diversity of these animals, look at population structure, then try to understand why we see these patterns, right? So first characterize the patterns, then ask, okay, what has led to these patterns in the first place? And then finally use this information to guide conservation management of the species. And so the first thing we're going to talk about is characterizing their genetic diversity. So how do we do this and what does it look like? So the first thing I want to show you is just, this is like a little, pretend you're all tourists coming along with me to Madagascar. How do we get to our field site in the first place, right? We probably all have wanderlust at this point after being locked away for the last year or so. And so we're going to take a trip to Madagascar. So the first thing you have to do is fly into the capital city of Antananarivo. So this is indicated here by the red dot. And this is like any other bustling city in the world. So you've got massive population densities, lots of hustle and bustle going on. From there, you hop in a car, a taxi Bruce, a 4x4, and you start driving south. And you drive and you drive and you drive. It's a 2-lane highway, one way in each direction. And back when the roads were really good, it would take maybe 8 hours. These days, it takes more like 12 to 14, but eventually you end up, oh, and I should note, it's not just the roads. These are what some of the roads look like. So these nice, muddy trails. Sometimes you've got to cross a river. The bridges aren't always there anymore. So then you have to build a bamboo raft. But eventually you end up in Ranomafana National Park, which is where I conduct a lot of my research. And so this is just this pull-out map here. This is Ranomafana. Here is where the sign is located. This is the entrance to the park. And then to get to my site, where the Ranomafana ruffed lemur project is based, is way the heck down here at Mangavu. So this is approximately 25 kilometers as the crow flies to get there, and there are no roads. And so instead we have to get a little bit more creative with our method of transportation. So the first thing you do is you take a dugout pirogue across the main Numuruna River, and you can see we hire 20 or 30 porters to carry all of our things, on our backs. This is a dry shipper with liquid nitrogen in there to help freeze our samples. So we're quite a sight to see. And after crossing the river, we just start hiking. So you hike and you hike and you hike, and you can, if you squint and look really closely, we're walking along ridge tops. Eventually there's a trail here, you go here, up over this ridge, and this mountain top way here at the back where the orange arrow is, that's where our field site is. So this can take anywhere from 6, sometimes to 12 hours to get there, but eventually you end up in this really beautiful, pristine primary rainforest site. And this is where we conduct our work. And it's sites like these where we conduct our work. The way we've done much of our collection historically is by using we dart animals. It looks a little scary. It's really not as scary as it might seem. So this is Velu. Velu's our darter. He uses a CO2 powered rifle to shoot darts that contain anesthesia in them. So we'll anesthetize the animals, bring them back to camp. And then these are a bunch of the different veterinarians that I've worked with over the years. This is Randy Youngie. I still collaborate with him at the Columbus Zoo. But at this point, we were able to process animals. And it's a great opportunity. I bring students out with me all of the time and volunteers, and we take photographs, we collect morphometric measurements, we collect blood and tissue samples, endoparasites, ectoparasites, anything you can think of. We then give them a radio collar and release them back in the wild to study them. But then what we have to end up doing is we take those samples. What do we do with the genetic samples once we have them in hand? Well, unfortunately, traditionally, we've had to bring them back to the States or bring them back to wherever it is the researcher is from. And this is because many primate host countries lack the infrastructure or funding to have labs. And this is something that my student Nuru ran into, is that she wanted to do genetics in Madagascar, but there just wasn't a genetics lab. So we had to figure out a way to get her over here stateside. But I just want to give a quick shout out because there's a new nonprofit called Mahalina, and it's run by Fidi Razumbainarivu. He's based in Madagascar. And what I think is so cool about this is that I guess one side is profit, one side is not for profit. The profit side is that Fidi is able to charge researchers to run their samples in his lab. And the profit that he makes from this, he then uses to help fund Malagasy students in their training and in their research. So it's a really awesome organization. I highly recommend you looking it up. But so I'm going to tell you, I haven't been able to do it because he opened it after COVID, unfortunately, so I haven't been. So I'm going to tell you how we traditionally would analyze samples. So basically you get your sample, right? So your blood or your feces or your hair. The first step is to isolate the DNA. So you use whole bunch of reagents and chemicals to basically get the DNA from the cells. You then take that DNA, you pick a specific part of the DNA that you want to study, you amplify it, and then you sequence it. So it basically, you read all the A's, T's, C's, and G's in your DNA, and that allows you to get a genotype, right? So the signature you inherit from mom, the signature you inherit from dad, and that spits out something like this. So I'm working with microsats, and so you just have these sequences of numbers. This first, so 233, 235. That's just the size of the fragment from mom, the size of the fragment from dad. Stick it together. That's one of your microsats. So this is essentially, not super exciting, right? It spits out, just like this Excel worksheet. But what's cool is that from that, there are a whole bunch of statistical packages and programs that you can use to look at genetic diversity, bottleneck events, population demography, so whether populations are growing or shrinking, whether individuals are moving between populations, all kinds of things. And so this is what we were able to do with the black and white ruffed lemurs. So we actually went to 19 different sites throughout Madagascar, the entire eastern rainforest corridor. And so these hashed lines here illustrate the range of black and white ruffed lemurs in Madagascar. And then the colors of these dots indicate their current subspecies designation. Okay, and so you can see that they sort of run the declinal variation from north to south, from subcincta, variegata, and editorum, all the way down in the southern region. And so what we expected to find was, in terms of genetic diversity and structure, that you would have clustering of genetics within each of these different subspecies, if they are in fact subspecies, right? And so what did we find? Is this the case? Well, the first thing we did was we took all that genotype data I showed you a second ago, and we ran it through an analysis just to plot them in space relative to each other. And if these are really good subspecies designations, you might expect 4 or 3 different clouds, right? A purple cloud, an orange cloud, a blue cloud. And instead what we found was one blue cloud, and then the purple and orange clouds kind of clustered together. And we thought that seemed really interesting, like a little bit unexpected. So then we did some more analyses. The next thing we did was something called a structure analysis. And basically this is a series of horizontal lines. For each horizontal line, that represents an individual. And the color of that line represents the proportion of that individual's genotype or DNA that the program assigns to a specific cluster based on frequencies within the data set. And so again, just like what I showed you a second ago, we see 2 clusters, not the 3 we expected. So we actually find a northern population and we find a southern population. And what's kind of cool about this is that what bisects them? Well, it's something called the Manguru River. And this actually aligns really nicely with other studies in Madagascar that have found shafakas and brown lemurs actually speciate on either sides of rivers. And so this actually makes a heck of a lot of sense. It's just not aligned with, current conservation units, as we might assume. Okay, what else did we find? We looked at another marker, the mitochondrial DNA. So this is something that's inherited just through matrilines, so passed on from mothers to offspring. And what you see, this is kind of scary, is that in the northern population, so these 2 groups here, you see a whole bunch of diversity, a whole bunch of variation, which is indicated by the number of black dots. And in the southern population, you see there's only 3 gray dots, which means there's a lot less diversity. And so when we're talking about this within population characterization, right, because that was goal 1, is just describe the variation, we see that in the northern cluster, so those green sites, there's a whole bunch of genetic diversity, which is great because that means adaptation is still possible. There's low FST, which basically is differentiation between populations. So that means if for every single pair of sites you compare, it's saying genetically they're pretty similar to each other. And there's no evidence of bottlenecks, meaning that population size hasn't rapidly crashed recently. And so this suggests sufficient gene flow. So those animals in the north are doing quite well. By contrast, in the southern cluster, there's very little genetic diversity. There's a lot lower just genetic diversity overall. There's high FST, meaning that for every pairwise comparison of sites, they're actually pretty different from each other. So almost 86% of them were different from each other. There's evidence of lots of bottleneck events, so rapid population crashes. And so this suggests limited gene flow and low adaptive potential in those animals in the South. Okay, so with that though, then we want to say, okay, well, why, right? Why do we see these different patterns? And so we want to understand these drivers of gene flow. And the way we're able to do this is using something called landscape genetics. And landscape genetics is this really amazing field that combines population genetics that I just described, along with landscape ecology and spatial statistics, to allow researchers to infer the dispersal ability of a species by examining the relative effects of different landscape features on the spatial patterning of that genetic variation I just described. And so essentially what this entails is taking those patterns of genetic diversity I just described to you. So for every single site, what does the genetic diversity look like? And then relate those patterns of diversity back to natural and anthropogenic features in the landscape. So it could be an ecological feature like forest cover or forest height, something like that. It could be geographic, like mountain ranges or rivers. It could be historic or demographic. So maybe these bottleneck events are happening or something like this. We know things were almost hunted to extinction, and also anthropogenic, right? So the presence of cities or agriculture, something like this. And so we look at all of the interrelationships among these different patterns to help explain the genetic diversity. And then based on how we, our understanding of an organism, we can actually make predictions about how difficult those different landscape features might be for an animal to cross. So for example, in a forested habitat, maybe it's really easy for animals to cross through a forest, right? That's what we would expect for ruffed lemurs. And so we would say there's low resistance to movement by crossing a forest. But what about if an animal tries to cross a river and never is able to successfully do so? Then we would assign high resistance values to rivers. And then in some cases, you see that animals can traverse low altitude and maybe mid altitude, but once you get to high altitude sites, it's just not possible to cross. And so then you have variable resistance values. And so by considering all of the features and all of the relative challenges they pose to individual movement, then we can identify suitable areas in the landscape or corridors that can promote and maintain gene flow and connectivity. And that's really the ultimate goal, is to figure out what is it that an animal needs to maintain gene flow so that we can maintain healthy populations. And so for our study, remember that we found that the population seemed to sort on either side of a river. So we thought, okay, well, maybe there's natural features driving gene flow. One is rivers, as I mentioned. The other is geographic distance. This is our null hypothesis. And basically all this means is that the farther away any two individuals are from each other in space, the less genetic variation they're going to share, right? So like if you and someone in your neighborhood live close to each other versus you and someone in, I don't know, Idaho, right? You might, if you're a naturally breeding population of animals, you're going to be more closely related to the animals that are closer in proximity to you. Okay. And then we also thought, well, there are elevational preferences for some species, so maybe we'll test that as well. And then topography. So here we can see this is rivers, valleys, and slopes. So maybe they're preferring to travel along ridge lines or something. And then we also decided to test anthropogenic features. And those included the presence of roads, right? Because we know that poses threats to many animals in terms of crossing roadkill, right? Habitat quality. And so we use this as a proxy for agriculture and habitat degradation. And then also distance to villages. So the more built up an area is, we figured that would pose greater resistance to movement. And so again, if we think back, these are the microsatellite data that we had. We generated that from those samples initially. We then generated genetic distances. And remember, I told you that the genetic distance is just a pairwise comparison of how many genes any two individuals share with each other. And so if you're completely genetically similar, your FST value is 0. That means there's 100% gene flow between your sampling localities. If you have an FST of 1, that means there's no gene flow. You're completely genetically distinct from each other. Okay, so you use the microsat data to generate genetic distances. And the first thing we wanted to know was, okay, well, is this just a isolation by distance? Is it just that we're less genetically similar because we're farther away from each other? And sure enough, you see that this is the case. 28% of the variation is explained just by genetic distance within that rainforest corridor. But that means that there's a good chunk of the variation that's not explained by distance alone. So this seemed promising. And so the next thing we wanted to do was test our hypotheses about all of these other landscape features that might be driving gene flow in the species. And so we were able to assign resistance values using this really new, really cool program. So traditionally, experts just assign resistance. So it would be me saying, oh, I know ruffed lemurs. They don't really come to the ground very much. So I think open areas of habitat would have high resistance. I'll give that a 10. And I know they like fruiting trees. So any place there's fruiting trees, I'll give that a resistance of 0 or 1. But again, that's kind of an arbitrary assignment, and it's really open to bias. And so this new program is cool because it basically is it's a machine learning program that will try and learn a path. And then with each iteration, the paths evolve to become more efficient and more parsimonious until you get like the best answer. And so what you can do is take the best fit surface for every single different landscape feature in isolation, and then compare all the different combinations to see which best explain the variation in the genetic diversity. And so again, you take those genetic distances, you combine them with the resistance surfaces, and what you get from this is this really cool heat map. This is from something called CircuitScape. So it's all based on electric circuit theory. So the way you would expect electricity to travel. And basically what you see here is that the warmer the color, the more conductivity or the more gene flow there is. And again, down in the lower portion, there's very little heat, there's very little movement, right? And as you move north, you get more movement, and that makes a lot of sense since the animals in the north had the higher genetic diversity, the higher gene flow. And so what we found is that the largest drivers of gene flow include forest cover, so the presence of good quality habitat, and human habitation, such that the more densely populated an area, the lower the gene flow. And this actually aligns really nicely with some distribution models that we did earlier, where we found that the best predictors of ruffed lemur presence using landscape imagery was the presence of forest. So really, these animals just need forest in order to survive. But so what does this mean, right? What do we do with this? Well, the first thing is we can look at it on a much finer scale. This is something my PhD student Amanda Mancini is working on right now. So rather than focus on the entire southeastern corridor, she's focusing just in this little area, but using the same exact sample size to see, okay, if you change the scale of the study, does it matter? And she's actually found that yes. So for our study, this broad scale study, we found forests matter, people matter. But on this smaller scale study, she found it's not just forests, it's actually the height of the forest. So they really like those really tall fruiting trees. And so if that's what's driving some of the gene flow, at least, is that it's only in these really tall stands of fruiting trees that the ruffed lemurs are actually able to move between patches. And then another of my PhD students, this is Aparna Chandrasekhar, and she's using coalescent theory, so lots of mathematical modeling, to actually use those same genetic data to infer patterns of population growth and expansion, and then try and link this to human activities. And what we found is, in fact, populations began to crash pretty much as soon as humans arrived to the island, unfortunately. So there's a pretty close link between us and their decline. Okay, so let's just do a quick recap then. So what do we know? We know that there's continued rapid fragmentation and loss throughout the eastern rainforest habitat in Madagascar. We know that in ruffed lemurs specifically, there's variable genetic diversity. There's more in the north, much less in the south. The animals in the south are in trouble. And we know that habitat quality and human habitation predict those patterns. So what we see is that the more remote, the more pristine the forest, the more gene flow, the healthier the population. And that as forests become lower quality and as they are closer to villages, then there's less gene flow and it's a lot harder to traverse the landscape. And so the final thing I want to talk to you about then is, okay, what do we do, right? What can we do about it? Because all of this isn't really super helpful unless we can use it to enact change, right? And so how can we use this to guide the conservation management of the species? And so as I noted in the beginning, we focused our attention on ruffed lemurs, one, because they're an indicator species, right? So they indicate the health of an environment, and when they're gone, we know the environment's not healthy anymore. But we also know that given their broad distribution, they have these really sort of specific habitat requirements, but they also are pretty widely distributed, and they're charismatic. They can serve as this really effective umbrella species, okay? And so basically what this means is that you can protect this charismatic, really cute, fluffy animal, but then the umbrella is that it also protects a whole bunch of other stuff that's maybe less charismatic, like some chameleons or geckos or frogs or leeches, right? That we might not otherwise be as interested in protecting. And so taken together, how can we use this information, right? So the combination of charisma and lots of knowledge about their ecological tolerances and their gene flow to turn what seems to be a pretty big conservation crisis into a conservation opportunity. And, it actually, well, it's hard to say. I was going to say it came at a pretty opportune time. So last year was the 60th anniversary of Madagascar's independence. And so in celebration of that, their president decided he wanted to have this big push to re-green Madagascar. And so in celebration of their independence, he planned to plant 60 million trees across 40,000 hectares in that year alone. And I'm sure that things have paused because of COVID, but by early 2020, he and his colleagues had planted 1 million trees in 500 hectares. So this is essentially almost 2 times the size of Central Park, okay? And there were an additional 100 million seedlings that are basically ready to go. They've been cultivated, they've been distributed, they're just waiting to be planted. The problem being that it seems, according to experts, according to scientists, there's really a lack of a coherent plan, right? So the idea is just to plant these things, but it's not really necessarily informed by science, and so we don't really know how effective it's going to be. And so we're hopeful that by offering suggestions that are empirically driven, we might be able to take this really, really amazing opportunity and more strategically target areas for reforestation. And I just want to give a shout out too. So this is Ed Lewis. He is at the Henry Dorley Zoo in Omaha, and he and his colleagues started a nonprofit that's completely Malagasy run in Madagascar called the Madagascar Biodiversity Partnership. And he is putting some of these results into action. So I've collaborated with him on some studies. This map here on your right is one of those examples where we did not landscape genetics, but population genetics study in this small forest fragment and identified areas where there was lacking genetic diversity, and then took the information we knew about gene flow to target reforestation corridors. And so he's done all kinds of amazing work with the local school children to get them involved in nurseries, and he's got a whole bunch of local villagers who are working to follow the animals, and he's doing amazing work. And his team, he isn't specifically, but his whole team is doing amazing work. And in total, they've already, with the help of Arbor Day Foundation, planted almost 3 million trees just in that one area alone. So I think it's this kind of grassroots organizing that's going to make a huge difference moving forward. And so this really, at the end of the day, my hope is that the results from these studies and others along with countless other studies dedicated by dedicated scientists working, all throughout Madagascar, can really help to guide conservation practice moving forward. So, for instance, our deforestation protections allow us to identify important climate refugia. We can highlight important corridors for gene flow, target areas for reforestation, identify important refugia that warrant increased protection. So again, this figure is from that forest loss paper, where we were able to actually target, with climate change and forest loss combined, areas that would still maintain suitable habitat for ruffed lemurs, and then really lobby to make those areas protected. So more than just paper parks, but actually get them enforced and protect these areas, and maybe even prioritize new areas for conservation. And then, sort of as a last ditch effort, if we are able to target those areas, then consider doing translocations and reintroductions into those areas. And I do have to say, with the help of local governments and amazing conservation champions in Madagascar and local communities, I really do think, it's hard to give these talks because it's hard not to just you feel like it's a punch in the gut, like, oh, what can we do? But I think we're moving in the right direction. And I think that there's a lot of room for hope. And so, at the end of the day, I think there's plenty of opportunities to really turn what many consider to be a conservation crisis into this really, really amazing conservation opportunity. And so with that, I just want to acknowledge colleagues and funders and students and my teams, both here and abroad.
Okay, so kind of give us a little bit of backstory here. How did you get started in this particular work?
I think that's a great question, and it's one I like to tell, especially to students, because I was actually I was a photojournalism student at the University of Miami. I had already bounced through several majors. I was elementary education, I was art photography, I was photojournalism, and I just decided I wanted to do a study abroad and do a photo documentary project on something. And once I got there, I realized I would much rather study the animals and learn about the animals, and it kind of changed my life. So yeah, I never knew it was a career until I tried it. And once I did, it sort of changed things from there on out. So yeah.
So when exactly were you in Madagascar?
So I first went, actually, it was sort of traumatic at the time, right after 9/11. So I was on the plane flying to Madagascar. We didn't find out because there were no phones, there was no internet. It took us 2 weeks to even find out that 9/11 had happened. So that was my first trip there as an undergrad. And then I've gone back. So that was 2001. And then I went back starting again in 2003. And I've been back every summer. So our summer, their winter since then. And then for my dissertation, I spent 17 months living there, actually in that rainforest that I showed you with the tent. That's where I lived for 17 months. Mm-hmm.
Would empowerment of soil by shifting agriculture creating a problem for planting and growing native plants?
Yeah, and I think that's a big thing to consider. And so one reason why lemurs are so important in particular, actually, is because by ingesting the seeds and passing them through their guts, it actually scars the outer covering of the seeds. And then also their feces acts as fertilizer. And so that's kind of like a perfect system for regenerating forests. And so actually what Ed does, so he and his team with the Madagascar Biodiversity Partnership, there are entire teams of people who follow the animals, collect the seeds from their feces, and grow them that way. And so I think that's like a really clever way to do it, because as Robin's suggesting, it's tough, right? You can't just go plant them on a hilltop. It's all red clay. Nothing's going to survive. So yeah, we have to be strategic in how we do this.
I'm not quite sure I would want to be the one to have that particular job, but it would be interesting. It's different. All right, another one from Robin. Is there a way to include local villagers in the solution? I
I
apologize
think
if you
that's,
covered this already.
yes. And I think that without the local villagers, there's no chance of it succeeding. So, that's something I learned early on. And I think it's so valuable, and this comes up again and again in conservation, is that you have to ask the villagers, what is it that you want, right? What is it that you need? And then let's work together to make that happen. Because if you don't have buy-in from local communities, uh, it's never going to work. Yeah. And so I think a bunch of my colleagues and I have tried to do this, and I work really closely with Malagasy students. So I bring students out every year. I actually have a Malagasy student, two PhD students that are Malagasy. So they're doing the training, and their goal too is to, once they've gotten their PhDs, go back into the communities and work against, keep the cycle going, right? And so I think that capacity building is the only way to go. Yeah.
And then that paired with, as you're talking about with the president of Madagascar, he is trying to introduce this great plan, and yet it needs to be backed with all of the science. So those two working really in tandem, I feel like that is, you covered that really well, kind of puts it into perspective for us.
Yeah.
And as Central Oregonians, we all know how important it is to be heard when you are in a place where a lot is happening. There's new growth, new development, and we are so closely connected with nature too.
Absolutely. Yeah.
So you mentioned that you go back every summer, their winter, to the same area. So how long has this project really been going on? How long will it go on? Are there any other projects that you're working on?
Yeah. So I really got the project off the ground in 2005. So it's been running since then. Unfortunately, the last time I was there was 2019. I was able to employ my techs to continue working until March, and then it just wasn't safe for anybody. So it's sort of on hiatus for now. But I think we've been able to get creative and sort of shift focus and work with some of the older data. But so just to give you a few, well, I'll describe my most exciting project for me right now is a study that's looking at those same lemurs, so the black and white ruffed lemurs. And they're cool because they have this weird infant care strategy. So they have two or three babies at a time. They're the only primates, day active primates that do this. So they're weird already. But what's really interesting about them is they build nests like birds. They park the babies into nests, and then moms will sometimes park their babies together into what we call kindergartens. And then moms will take turns caring for the babies. And what we found is that through that communal infant care, it increases infant survival. So not all moms do it, but the ones who do have babies that are more likely to survive. And so we've already learned this from previous studies. But so now what we're interested in is looking at the gut microbiome. And I feel like this is a hot thing right now, right? If anyone's taken probiotics to improve your gut health, right? And it relates to health and immunity and all these things. So we're actually looking at moms, ruffed lemur moms, who don't have help versus moms who do have help, to see how the baby's gut microbes develop through time, and then relate that back to patterns of social relationships, to genetic relatedness, to whether their kid, like how social their parents are, and then long-term patterns of health, to see if we can understand, how our social relationships and our social environment helps to shape gut health and then our own like long-term health, which I think is pretty cool. So that's something I'm working on right now.
That's amazing that you can do that from afar too. And
Yeah.
is there anything that would be indicative then of whether a mom would choose to kind of do the kindergarten thing? Is there anything that you found so far that would perhaps give us a clue into which ones might?
Yeah, so it seems like it's friends and it's relatives. So moms who are outside of mating, outside because they have seasonal breeding, so they only have babies in October. That's it. And so outside of that October, it's the females who have long-term friendships. They're more likely to cooperate. And then it's also the females that are close relatives. So like mother offspring or mother daughter or siblings will help.
So how does that work in with the lemurs being kind of territorial? Is it just within those large groups, or are the males the ones who are more territorial than the females?
So it's cool actually. So ruffed lemur, all lemurs, females are dominant to males. So it's the females who defend the territory. Males are really submissive. Females will sort of swat at them, beat them up a little bit. And then, yeah, so for the care, it's only within that territory. So within a territory, usually you have like 5 to 8 adult females and about equal numbers of males. And so it's those females that'll care for each other's kids. Yeah.
That's fascinating. I love
Yeah.
that.
Yeah.
Okay, and so because the environments are getting encroached on, you mentioned that a couple of different solutions or stopgaps would be translocations and
Mhm.
relocations. Can you tell us exactly what that would entail?
Yeah. It's widely debated. And I feel like from my point of view, at least, it's kind of a last ditch effort if we had to do this. Um, but what's happened in the past actually is if you identify a population that has low genetic diversity, but it seems like the habitat is fine, it's good enough quality habitat to support a healthy population, then what you can do is one of two things. You can take captive bred animals. So Duke Lemur Center has actually done this, where you can breed animals in captivity, bring them, sort of train them how to forage, and then release them into the wild. And fingers crossed, hope they mate with the animals that were already there to increase genetic diversity. Or you can identify an area where the forest is likely to be lost. So, for instance, this has happened. There are massive mining operations throughout Madagascar that just clear cut forests to get to the resources beneath them. And so if you know a forest is going to be cut down, you can take those animals from that forest and translocate them into a population that maybe needs more genetic diversity. A few people have done this actually successfully. So they've done it with shafakas, they've done it with ruffed lemurs. And so I think, at some point we're going to run out of forest. So we can't, that's not the only solution, but it seems like it's working. So it's another option.
Andrea, is there anything else you want to tell us? Anything else you're just dying to say? Anything you want to share?
Gosh, I don't know. I I say you never know what you're going to like until you try it. So, I think we should all be a little bit adventurous in terms of just giving things a shot, because really doing that changed my life, and for the better, I think. And then I do also think, just doing our parts. Granted, there's giant corporations that are involved in lots of these, deforestation and things like that, but just being wise consumers and really knowing where our products are coming from and trying to be responsible with how we, who we support, because money talks. And I think that's something I always try and remind myself of too, is just, put your money where your mouth is. And we can't all do everything. And so, if you don't want to make sacrifices in certain areas, maybe you can make sacrifices in other areas, because I think in the end, collectively, whatever little things we're able to do can make a difference.
Wow, that was very well said. Thank you so much, everyone, for joining us tonight. This was Endemic Animals of Madagascar, part of Deschutes Public Library's virtual programming. We have so much fun free content for you available at our YouTube page. So please go and check us out there. And of course, our libraries are open, so come see us in person. We have curbside pickup and we have lots of other services, including our mobile app. So check it all out and we hope to see you again soon. Andrea, thank you so very much for joining us tonight. Happy weekend. We hope to see you all again very soon.